to introduce both this work as well as Zach. I will speak a little bit more about both, but I just wanted to, take a few moments to, really, this is a wonderful set of findings that we'll walk you through. So these are some of the methylomics data that were generated, of course, from the blood sample donations that our families have provided. There is going to be an additional wave of these data and more opportunity to contribute that Zach will talk a little bit more about. So these are some exciting initial findings, but we are really dedicated to having all of our 8P heroes represented here. So if you haven't donated blood, or we'll talk a little bit about the subgroup of the population that were kind of captured in the final analysis here. If you're not in that subgroup, we definitely want to extend these analyses. So the methylomics data were then evaluated following their production at Illumina in partnership with an incredible organization called EpiSign that specializes in making these epigenetic signatures for different conditions. And so they've done this for a wide range of conditions. Zach will talk all about this, but they've now done this for 8P. And so this is something we're publishing in collaboration with these groups. And we've learned a lot through the epigenetic signature that they've made, which really helps us to contextualize both 8P within kind of the disease landscape more broadly, but also to understand more mechanistically about 8P and what opportunities that we have to develop interventions. So this is really exciting work of a lot of folks coming together. Maybe we'll go to the next slide, Zach. And I think, oh yeah, so we are recording this. Yeah, indeed, we're recording this. And so if you prefer not to be included, just send us a private message and we'll edit you out. And then as usual, we won't put the discussion in. We'll just put the presentation section in. And then do I have one more or one more? Okay. Beautiful. So as we go through, as you have questions, we, especially since it's a smaller group today, please continue to put your questions in the chat and we can navigate them as we go or at the end. But this is really some exciting work that I'm excited to dig into and want to make sure that everybody understands. And the person who will be guiding us through that understanding, Zach Gershon, we're so grateful to have him with us. So he's just finishing up like 2 months. He's a little bit over now. And so he is our scientific program manager. He just completed his postdoctoral fellowship at Rockefeller University. He has a PhD in neuroscience, specializations in genetics and cognition. He's been hitting the ground running with us and, bringing some order to the 30 plus collaborations that we have rolling now. So he has just been absolutely phenomenal in helping us get all of our ducks in a row and using all of the beautiful data that has been generated over the many years that the foundation has been, working so hard on 8P. We're getting to a really beautiful point where we're able to leverage a lot of that early work to have some exciting findings that then we can continue to build on. And this is a really great example of that. And so with that, I'll hand it over to Zach. And yeah, we're so thrilled to have him. You'll see much more of him. And, please feel free to reach out to him after with any questions directly as well that you may have in follow-up to today's session. But thank you. Go ahead, Zach.
Thanks, Lauren. I just want to let you all know how excited I am to be here at such an interesting time in all of the science that is being done within the 8P community. So for some of you, this background will be a refresher, and for others, it'll be new. Like Lauren said, if you have questions, please feel free to put them in the chat or reach out to me or Lauren afterwards. So everything our cells do is carried out by the proteins they make. Genes are the DNA instructions to make each protein. The process of reading the DNA instructions into RNA, called transcription, and building the proteins from the RNA message, called translation, is a process known as gene expression. While epigenetics are processes that regulate gene expression without changing the DNA code. This is a normal part of how our cells function. And there are many types of epigenetic mechanisms. One important mechanism is known as DNA methylation. This is when a small mark called a methyl group is added to specific DNA sequences. These methyl marks make the DNA they're on wind tighter, essentially making it difficult for the machinery that carries out the transcription to attach to the DNA so it can read and transcribe the code into RNA. While these methyl groups are on the DNA, they don't change or disrupt it, allowing for flexibility of this process to be reversed. You can think of it like the cell putting sticky notes on your DNA, telling it which sections to pay attention to and which to ignore, which can then be removed later. It's a pretty efficient way to prevent the expression of genes that shouldn't be expressed and prevent the transcriptional machinery from landing on regions of DNA in between genes. That's why DNA methylation plays important roles in critical biological processes, including brain development and rewiring. During development, changes in DNA methylation help regulate genes that direct the rate that stem cells in the brain grow, how often they divide, and what kind of cells they end up becoming. It also helps control how new brain cells get to where they're supposed to be and what other cells they connect with. Additionally, DNA methylation is involved in guiding how neural pathways change or how they're strengthened to stabilize important existing connections. This is very important for cognitive processes like learning and memory. Because of all of the processes DNA methylation is involved in, it's very normal for us to acquire more methyl marks on our DNA as we age. This means that cells divide more slowly, don't repair themselves as well, and work less efficiently overall. However, when epigenetic regulation is disturbed, cells can display signs of advanced aging. There is evidence that this happens in Down syndrome, which I'll talk about a bit more later. But this is one of the reasons we thought DNA methylation was worth looking into. Especially to understand how epigenetic regulation is disrupted in chromosomal rearrangements because it can lead to increases or decreases in epigenetic regulatory proteins, increasing the distance between the transcriptional machinery and where it needs to go to start the gene expression process. And if the surrounding sequence is different, the DNA containing important genes can be wound more tightly, preventing expression of those genes. So, understanding the epigenetic state of chromosomal rearrangements is very helpful in understanding important pathways that might be disrupted. Because of this, as a foundation, we collected methylomics, which is looking at all of the methylation across the genome and how it might be driving symptoms in 8P. We're integrating this and all these different layers of biologies listed here on the slide, which are measured in what are called omics, so we can best understand the biology of our heroes. This methylation analysis is from the blood you've donated, as Lauren mentioned, and we're going to be expanding this study with further collection in 2026. Unfortunately, our current epigenetic analysis had to be restricted to indupdel because the sample size for other rearrangement types. But we will expand to all the rearrangement types next year, again, following additional sample collection. So to assess the DNA methylation of the heroes, we sent the blood samples to Illumina, where scientists extract DNA from those samples. A machine then scans the DNA and measures where the methyl marks are and how many there are. That's the methylation profiling part. Based on hundreds of thousands of reference data points, a computer decides which methyl marks are the most informative for identifying a unique pattern. Then machine learning is used to evaluate that pattern against all other disorders EpiSign has assessed. Finally, a unique episignature for that disorder is identified, which we can then compare with others, especially other chromosomal rearrangements. But why would we want to compare epigenetic signatures with other chromosomal rearrangements? Well, disrupted genetic information causes some kind of stress on cells, and we want to know if that's driven by changes in epigenetic regulation, as well as if those changes are shared by other instances of a genomic imbalance. It will also let us understand what biology might be shared with other rearrangements and build off the work that's been done in those that have been studied for a long time, like Down syndrome, basically allowing us to stand on the shoulders of giants. So this diagram shows how similar the epigenetic signatures of all of the disorders EpiSign has evaluated are. In the diagram, the distance of the lines connecting each circle, which is each circle is a given disorder, represents how similar the disorders are. So those that are closer together are more similar to each other. The size of the circle represents how many unique differentially methylated regions, or DMRs, are in that episignature. And the color represents how much methylation is in each disorder, according to the color bar here at the top, where more red in a circle indicates more methylation or hypermethylation, and blue indicates less methylation or hypomethylation overall. Now, going into this, we hypothesized that 8p would be most similar to other chromosomal rearrangements like Down syndrome because of excess genetic information. However, chromosome 7 duplication is all the way out here on the right, and is hypomethylated, while indupdel is close to Down syndrome, and both have a more hypermethylated signature. So while we were correct that indupdel and Down syndrome would be similar, it doesn't seem to be a feature of other chromosomal rearrangements overall, which suggests that it's not necessarily driven by excess genetic information, but rather the similarity in epigenetic signatures is likely due to similar changes in DNA methylation. And if we look at the overlap in important methylation sites depicted by the Venn diagram here.
Sorry, Zach, could you go back to that slide for just like a couple seconds?
Sure.
This is like part of the meat of it, and this is part of what's going to be in the publication, which is really cool. So this is showing our epi signature on the map of all of the different diagnoses and conditions that EpiSign has looked at. And then Zach took like some snapshots to help us understand. So there's like more conditions that are further away, but the closest that it is to us in this diagram is showing like of those different methylation sites where they are in the genome, which conditions we most resemble. And our hypothesis, because I'm sure you've seen these data, like even at the last Family and Science conference, that we had overlap in the transcriptome, so that like step before the proteins with Down syndrome. And we thought it was like all chromosomal rearrangements, or perhaps just the ones where you have like these big duplications, but that's not what we saw. So there were other conditions that they looked at, including this like big duplication on chromosome 7. You can see, I don't know, Zach, if you can hover your pointer, but that's that one, and that's really hypomethylated. And there's a huge number of sites that are included there versus like there weren't as many sites for us, but we were primarily hypermethylated. So it's like overly simplistic to say it's like opposite, but like it was not the same across all chromosomal rearrangements that have this like big excess of genetic information that's happening in the nucleus. And we kind of before this thought that maybe there were similar pressures for all of these. So like it's a very, very interesting thing for us. And then again, the fact that we're still showing up here as similar to Downs, we'll talk a little bit more about this, but like it's very interesting for us to start thinking about more and more of what we can leverage from our understanding of Downs because there's so much research that's being done in that area. It's like a juggernaut. So it's like easy for us to tap into, even though of course there are key differences with AP. I'm stealing some of Zach's thunder, but this is just so exciting. I wanted to just like pause again on that figure and just, really help us with that. But yeah, so we'll see it again here and but yeah, so this will be in the publication with that EpiSign group. And the other cool thing, the last thing maybe I'll say about this figure is this is a permanently living figure now. So as we go through and we add more 8P groups, that we add our the deletion group and the duplication only group, and as we start to add more and more, and as EpiSign starts to add more and more, including other like CNBs, other chromosomal conditions, we'll see where we line up with those other conditions. So like this is a very wonderful living, relationship that we'll continue to have with this group, and we'll continue to learn more from being able to integrate our epigenetics into this resource where their goal is to be able to diagnose all conditions based on epigenetics, which is, really a neat thing to think about. But anyway, back to you, Zach.
Yeah. Um, the overlap of important differentially methylated regions between Down syndrome and the indupdel 8P. Um, we see that there are a little more than 1,700 shared important methylation sites. And this, relatively small amount of overlap compared to all of the important methylation sites in that epigenetic signature is actually really helpful to us because it helps us narrow down on the more likely important differentially differentially methylated regions that could suggest the shared biology that we see in Down syndrome and 8P. We've also seen, as Lauren mentioned, similarity between the indupdel and Down syndrome in differential gene expression. And this adds further support to that overlap. As a foundation, we have several ongoing projects about this, um, which uh, stay tuned and we'll be talking about at some point in the future. Um, so, what can we learn from this shared biology with Down syndrome? Well, eventually we can share the therapeutic and intervention opportunities if there is sufficient overlap. However, this does not mean that 8P is the same as Down syndrome. We, in fact, know conclusively that 8P is more different than it is similar to Down syndrome. And I'll refer back to this slide for that. There is a small amount of overlap um, as opposed to what is defining that signature overall. The overlap does appear to be causing shared disease processes. Um, and using this shared biology, we can accelerate our impact for 8P heroes by understanding more about how we can work together with the broader chromosomal community to improve the lives of our 8P heroes. Essentially, we want to embrace their successes and avoid their mistakes. So looking ahead, we will be repeating this analysis for other 8P rearrangement types with sufficient sample sizes. So we're so excited to expand sample collection in 2026. We're also going to assess the differentially methylated regions in indupdel to see which biological processes they're involved in and how those markers affect DNA packaging, whether it's wound super tight and inaccessible or if it's open and allowing more gene expression. We'll also be evaluating the overlapping differentially methylated regions with Down syndrome to see if there's any overlap with the differentially expressed genes identified by Dr. Scheltzer and Dr. Meherena. We're also planning on exploring other epigenetic mechanisms that work hand in hand with DNA methylation, but can impact multiple genes at once, including mechanisms that influence how chromosomes interact with each other. Before we turn it over to questions, I just want to take a second on a personal note and say how glad I am to have the opportunity to join this community and take part in such important work. So thank you for having me. Now, Lauren and I are happy to take any questions you might have.
Oh, thanks, Zach. Thanks everyone for your attention. Yes, please come off mute or put questions in the chat. Really excited to hear what everyone thinks and has to say about this.
Thanks so much. I'm sorry, I'm just going to jump in. I've just come upstairs because you actually kept Madison's attention for the full 30 minutes, which was amazing. So thank you very much. That was a really engaging talk. Yeah, and really exciting data and seeing how close we are to Down syndrome. I think my main question when I looked at the hypo versus hypermethylated is like, what do we know about where we have, especially the duplication, about would it be HDACs? I can't remember which ones cause the methylation, etc. Do we know if there's sort of lots of HDACs in that uh, methylated bit, or sorry, in that duplicated bit? Do you have any idea what I'm talking about? Sorry.
I do. So the histone acetylation is the other like form of epigenetics that we were kind of alluding to at the end there. And so we are looking to analyze that as well, but that's a different assay than this. Um,
Okay.
but it's another one that we're really interested in exploring.
Okay.
Um, but yeah, Zach, do you want to comment any more on that?
Faye, were you talking about the DNA methyltransferases?
I think that's what I'm trying to get to. So like I'm thinking where we've got this duplicated region, obviously we've got more gene being expressed, or we presume we have more gene being expressed, right? Because there's 3 times ploidy. So I'm thinking, is it just that there's more, is it histone methyltransferases? Is that what it is? Is that what I'm just not remembering in that region? And I can't even remember how many there are. I just, my recollection of all the epigenetics, what happens in epigenetics is really quite poor. So,
So as far as I'm aware, the there are like 3 or 4 DNA methyltransferases, and none of them are located in 8P. So this is some sort of process that, the rearrangement
somewhere
of
in the
8P
pathway.
is, yes, is would likely be affecting and accelerating the
Mm.
DNA methyltransferases activity.
Okay. And do you think that's the only kind of explanation for it? Or do you think there's other like factors at play that just haven't hit my mind yet?
That are causing the hypermethylation.
Yeah.
Yeah, so there are likely, a variety of things at play here. And so the good news is there's a lot of research being done on this. So there's a lot of work we can do to try and understand how that's coming about. But this sort of second layer of how like the histone acetylation on the chromatin
Mhm.
so is affecting the chromosome like more holistically rather than like individual gene sites. That's like another layer of analysis that we're looking at as well, and that could help explain more of this like higher level why we see differences in gene expression kind of across
Mhm.
both the chromosomes. And that other piece that you touched on is another important one that we're doing in 2026, but it's not just what genes are modified, but it's the genes that are being expressed and how that change and the number of that gene is affecting the amount of protein that's being made and the transcription layer between that. So like
Okay.
when you kind of follow that through the biology, like what's happening. So that's kind of another important piece that
Layer.
we're
So
doing.
perhaps like what you're saying is, where we do have the duplicated region, there might be more methylation as well to kind of try and silence some of it, like a sort of, natural regulation.
Yeah, but we're seeing it across all of the chromosomes that there are differences.
Okay.
And that's part of what's so interesting, right? Is like,
It's not just silencing the duplicated area. Yeah.
yeah. And and that's sort of getting at, like we were saying, like this bigger question of, is there something about having that excess genetic information that's causing like these changes across all of these different genes. And so we do have some of those genes that are shared now
Mm,
across Down syndrome and AP, even though it's two separate genetic regions, right?
yeah.
So, yeah. So there's another approach that we're also taking that's called high C, which maps like the chromosome interactions in space. So you kind of get like the 3D architecture. So you
Yeah.
start to untangle why a rearrangement in one area would be affecting these other areas that aren't like they don't have that change, right? But they're interacting
Yep.
together. So we'll learn a lot from that too. But yeah, you can start to understand then when you start to see disruption in certain pathways, whether or not that's relating back to the fact that there is this differential methylation, this stop go light kind of thing of whether or not that gene can be transcribed, and whether or not those are related. So there's a lot of really exciting like implications for knowing this, that not only is the rearrangement acting on, chromosomes other than just eight, but there are some important signals here that we can further understand.
Mm. That's great. Thank you. Really, really interesting. So thank you very